Both have different pros and cons, but as far as we see it, magnetic bead technology, due to the fact that we can integrate clarification and capture, has quite a large advantage, especially if you are in a cost-competitive market.
Hey, smart biotech scientists! We are in the middle of a conversation with Nils Brechmann, who is the marketing manager at MAGic BioProcessing and co-inventor of the process applications. And we are talking about ways how you can combine clarification and capture into one single step and a lot more. And if you haven't listened to part 1 yet, please go back. And today we are continuing our journey. But before we do that, what do you think about previous episode? Please tell us by leaving a review. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. If now we zoom into the physics and the chemistry involved here, can you explain how the magnetic separation works in detail?
From a ligand point of view, it's very similar to protein A column chromatography. It's the protein A which binds to the FC region. But I think what's a little bit more of interest is that you have, in essence, two different kinds of working mechanism, especially if you compare it to column chromatography. The first main difference is that magnetic beads, they cope with the particulated liquid, while column chromatography needs to have purified liquid. But taking that aside, it's still two different working mechanisms. While magnetic beads, they are more or less working in a stirred tank bioreactor. And then on the other hand, column chromatography, which is more a plug flow or tubular bioreactor. So therefore, the physics and the kinetics differ a little bit, even though on a small scale, the kinetics remain the same. I mean, there's no difference between the protein A's. But the magnetic separation has the advantage that you can use them in a batch absorption process, which means that all binding sites are presented the second the target actually hits the magnetic beads. So potentially what that means that all your target can bind basically the second both liquids are combined. While if you look at the column chromatography, obviously you have different plates or theoretical plates throughout your column. So only a fraction of your binding sites are presented at each time to your product or to your target. Both have different pros and cons. As far as we see it, magnetic bead technology, due to the fact that we can integrate clarification and capture, has quite a large advantage, especially if you are in a cost-competitive market. Just to summarize, to actually visualize how it really works, commonly at the end of your cell culture, you transfer the beads into the cell culture, or you can do it vice versa as well. You can transfer the cell culture into a bead reactor as well, where then the system is stirred. Obviously, the beads, they need to be in active stirring. Otherwise, they have quite a high density, so they will settle quite quickly. So therefore, active stirring is required. Once all the target has been absorbed, usually an external magnetic field is applied, which then leads to the effect that the magnetic beads are magnetized. They accumulate and they stick to the magnet. At that point, you can wash out all cells, all the debris. Once the magnetic field is removed, those beads are released as well, and the magnetization is gone more or less. That's about it. It's not more than that, to be honest.
And what are the current scales you can treat Are we talking small scale or are we talking even large scale bioproduction? Where are you at the moment?
Let me circle back a little bit and try to first explain the difference between other beads on the market and the ones that we are advocating. So currently the magnetic beads, you have large suppliers like, for example, Miltenny or Thermo Fisher, which provide beads that are rather small, 1 micron or even smaller. And those beads, they are actually constructed in the way that you have a magnetic core, and then outside of that core, you have bead construct built up where then your ligand is attached to. This unfortunately has the effect that the level of magnetization on your beads is rather low. While for our beads, they are much larger in size, 100 times larger, so around 100 micron on average. They are actually constructed the other way around. So we start with a base material, and then the magnetic material is incorporated into that material, which then actually leads to the fact that you can reach a much higher magnetization. And that also leads to the effect that you can use our beads in larger scale as well. Basically, it has the effect that the distance between the magnetic field and your bead can be much larger than for the smaller beads. The scales we're currently talking about, we have a study published where we looked into pilot scale, where we have processed up to 20 liter. And we have a prototype currently under construction, which is able to process 2000 liters. Obviously, it depends a little bit on the concentration, but overall, it's about to hold roughly 40 liter of beads.
And is there a limit somewhere, or you would say eventually you would even be able to scale it up to larger scales?
That's actually the nice thing using permanent magnets. There you're a little bit freer to do the scale up. To my opinion, you can scale it up even further, but 40 liter is currently the mark that we are looking at. Obviously, if you want to go higher in scale, then you have to do some adjustments, but when it comes to permanent magnets, that's something which is a little bit easier done than, for example, with electromagnets.
Now, speaking of scale, what comes into my mind is the cost, because obviously if you scale to larger scales, you will need more material and you obviously will cost more. Compared to Protein A, the traditional way, and the number of cycles you can use typically a Protein A column, is this comparable, or are you cost-wise much lower, higher, or where are you?
That's a very good point. Especially Protein A is still the highest cost factor in the downstreaming process. Currently, what we are looking at in the price range, we are very similar to the market leader in column chromatography resin. Same with the reusability. So we are on the same level as you would buy from other market leaders. However, obviously, it's a little bit difficult to only look at the Protein A because, we are not only saving costs in the Protein A, but we're also saving costs in the steps before and the clarification steps. So this, I think, is something that should be accounted in it as well.
Yeah, absolutely. I agree. As we highlighted before, you're saving an entire step. That's also a huge saving. So how does it tie into the general tendency of process intensification? Can this be used, let's say, in a more continuous way, for instance, for a perfusion process upstream? How would that work?
Generally, the answer is yes. I want to try to explain that a little bit and show also the advantages and also where other methods can be a little bit more beneficial. Obviously, the biggest advantage is that our system can handle cells. So therefore, if we talk about process intensification, I think most interesting are process intensification that still have a single harvest, for example. If we talk about intensified or concentrated fed batch process, where you reach really high cell titers and really high cell densities, those are extremely challenging for the centrifugation and filtration step. And I think there our system has a very high advantage to the unit operation steps that are currently used. If we talk about very traditional or very classic perfusion process where you have a membrane with a cutoff that is big enough in order to let your product perfuse through, then I think column chromatography might be a little bit more favorable. But if we only look at the actual setup, magnetic beads can be very easily set up in a similar way as you would, for example, use a periodic countercurrent chromatography system where you basically frequently cycle smaller batches after each other. That's something which can be easily done as well. But then on that same note, I think if we talk about the implementation of magnetic beads, it's much more interesting to use them on a non-clarified cell broth than something which has already been more or less clarified through a holofiber or filtration device, whatever you use in your perfusion process.
So what are then the main considerations we should take into account when we are thinking of adopting magnetic separation technologies, when we're developing a process?
To be fair, it depends very much on the upstream part. As I said, if you have a high cell density, single day harvest or single harvest process, I think then magnetic beads is something which should be highly interesting for you. If you still have classic fat batch applications or fat batch cultures, then as well, I think essentially it comes down to the fact, do you have a single harvest that still contains cells? I think that should be the primary consideration. On the other hand, perfusion process could be interesting as well, but there also column chromatography has the right to be there. So there is a little bit more diverse. But to summarize it, essentially, if you have a single harvest that contains cells, I think then magnetic bead technology should be something that's interesting for you.
Let's look forward a bit. Let's say in 5 or 10 years, we're having the same conversation as we're having today. What do you think will have happened in this exciting space?
I think, first of all, maps are here to stay. Obviously, the map market will continue to develop. Obviously, it's very interesting to see the different derivatives of full-length antibodies, talking about nanobodies, talking about different fragments. I think those targets and modalities are very, very interesting. And then for me personally, I hope to see that the field of cell therapy starts to really pick up and develop further. And then also AAV production and viruses, I think that's something which is really going to be hot still in the next 5 to 10 years. Then we really need to deliver methods that actually can help with the production and cope with the challenges we will find there.
And how will the magnetic bead separation evolve?
That's a very good question. I think that's something the shareholders would like to know every time as well. Well, we'll see. I hope that it can develop in premier methodology for the purification of cells. It still is for the small scale, but I hope that it can really have a big impact on the large manufacturing of cell therapy products. I really hope that it shows to have an impact in the AAV production as well. I think a lot of people are really curious about it. There are some nice studies that actually highlight the benefits of magnetic bead separation, especially if you have to deal with the cell lysis before and you have to deal with a lot of impurities in your cell broth. And then personally, I also hope that we as a company can also establish a foothold in the map market as well and try to show and make people understand that this is really a technology that can benefit the manufacturing process.
Nils, what is the 20-second takeaway from our discussion?
Be open to new technologies. I can even shorten it much more than that. Be open to new technologies. And there are many, many good technologies, and always try to start from scratch and be open to new technologies. That's my takeaway.
Fantastic. Nils, where can people get a hold of you?
Commonly LinkedIn, Nils Brechmann. That's probably the easiest. Otherwise, my email, nils@magicbioprocessing.com. That's usually how people can reach me. And I'm always happy to discuss magnetic technology and also discuss critics about it. Obviously, there are always pros and cons to all technologies. So it's always fun to also discuss with the other side.
Yeah, smart biotech scientists. I will leave all the links down in the show notes, and please do reach out to Nils and have these thought-provoking conversations about magnetic bead separation. So thank you very much, Nils, for sharing all the insights, the passion about downstream processing, developing new technologies, and it was a huge pleasure having you on the show today.
Thank you. It was so fantastic talking to you.
Hey, smart biotech scientists. Thank you so much for tuning in today. What did you think about today's episode? Please tell us by leaving a review. And perhaps you're wondering how you can master your bioprocesses. If this is the case, we want to make this very simple. I want to offer you a free 30-minute assessment. Just book a call using the link in the show notes, and I will help you get started because I want you to be able to master your bioprocesses. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.